Thank you, Prof. So, I think I have 20 minutes. I will just quickly go through. So, just a little bit about the outlines of this presentation. So, going to talk a little bit about the experience in drug discovery that we have done previously, mainly on the nature products at the beginning. Then later on, we'll talk a little bit about the challenges. So, during that process of the drug discovery, what are the challenges that actually stimulated us and came up with the ideas to come up with the additional initiatives, including the drug delivery to improve the activities, then also the cell line development inside our center for chemo-resistant study, then leads towards the extracellular vesicles research, which is relate towards the use as a biomarkers and also the treatment as well. Then we'll spend a little bit time. I need to do some marketing for Sunday as well. Yeah, so I have no choice. So I add in a little bit more about our center, single cells technology that we have invested. I think, correct me if I'm wrong, so 1.5 million Malaysian ringgit. So first machine in Malaysia. So my staff correct me, I thought it's 1.3. She keep on saying 1.5 from Go. Yeah. So then after that, I'm going to talk a little bit about our initiatives as well in terms of our multi-care pharmacy innovation hub on the clinical trials and everything, which is like ongoing at the moment. Yeah. So let me quickly go through. So myself as a biochemist, I was trained in nature products. And through this nature product discovery process, basically I was given the opportunity to pick up the knowledge in chemistry and biology. So with this as a fundamental, we do understand the biochemistry is fundamental for everything, living things, right? So from there, I expand my expertise and moving forward for different kind of the research, which is later on you will see, yeah. And initially we work with plants, then we worry about the sustainability. So we cut the plants to get the compounds for the testing anti-cancer effects and so on. But if let's say the compounds or your extract seems to be effective, you might have a problem because you need to keep on planting the plants. If let's say your plants took 20 years, like for example, just now the sky fruit, 20 years to grow and they start fruiting, then we are, I don't want to say that word, okay? Yeah, so that's why we started to think, can we ferment the bacteria start to synthesize useful metabolites. You can, from the bioreactor, keep on harvesting. That's why I started to touch a little bit about the Streptomyces, working with, I think, Prof. So microbiologist during that time. Yeah, and he's still, yeah, now in China. Then from there, we work with the different novel species. One of it is like, named after Malaysia. So it means that nobody else has found it. Then we named it after Malaysia. It's a novel species. And it was like 20 essential medicines listed by LT8 during that time. Yeah. And our student, I think Dr. Sir, now is a senior lecturer in Sunway University as well nowadays. And during that time was her project. She received the best award from the pharma companies from Taiwan. Then through all the way, received awards from Deputy President of country. Something like Malaysia is Deputy Prime Minister, in Taiwan is Deputy President of country. She received the awards from. So because of that discovery. Well, highlighted in newspaper, skip through, not important, right? Yeah. So a little bit of background. So I initially spent time, I think 10 to 11 years in Monash. Yeah, I'm still at there. So I think I've seen quite a number of colleagues are here, my previous colleague, that I'm so happy. Yeah. So during that time was a biofunction molecule exploratory research. As a small group, we worked with the project, eventually invited to join Sunway University. So a small group become a research center. So in research center, I think those who attended my talk before, they have seen these pictures many, many times. Yeah, so I always ask the black or white cat is a good cat. Why I'm putting this? As long as it can catch a rat, it's a good cat. So why I put this? So we work with the compounds, we always seek for its function. So if no function, no activity, it can't kill cancer cell for this context, no point. You can purify, you can synthesize, no point, right? But today, I'm going to switch a little bit of focus. If let's say we put this photo, then you start to treat, let's say we have a compound, we treat on the cancer cell. But most of the time, nature product activity is quite low, mild. Or you synthesize a compound, also mild the activity. Or initially, 72 hours able to kill, but with prolonged treatment, the cell form resistant. This is like majority of the time, the challenges we face, especially those who work with the synthetic compound or you are isolating it. So you can spend a lot of time, you found the compound but no activity or mild activity, you can't move forward. So how we actually overcome it? So working with the chemical engineering group, so we work with the nanoparticles as well. So one of the potential initiatives, so we work with the nanoparticles and we also look into the context of drug delivery. So we leverage on the expertise of our collaborators. So this is just an example. It's still a sky fruit, but we work with the nanoparticles and delivery to improve the activities. So supported by, I think now he's also graduated already, so it's a Dr. Darren. And we also work with the delivery way, but it's the magnetic stimulation release. So you can do a targeted release. So it was done by Dr. Low during that time, quite some time ago. So we culture the spheroid as well. Then we do the encapsulation, then using a magnetic stimulation to open and release. So those are the ways that we have an additional way to improve the efficacies of the treatment. Yeah. This is just like additional information how the thing is being stimulated and released. Okay, this is another PhD student that currently registered in Monash, so I'm the co-supervisor for. So we working on the emulsion way for delivery as well, and this is just accepted recently, 18 June. Yeah. So he did a review and we're actually working on it at the moment. And besides the drug delivery concept or nanoparticles to improve the activity, to give additional value to the research that what we're actually doing, in our lab, we use adenovirus to synthesize the cell. So the concept is simple. Most of the time chemo drugs, I think the reality in clinical setting is a combination. So from scientific research, if let's say your compound's activity is not too strong, you're also thinking about synergistic effect. But in real life, when you want to do experiment, you might face problems. So many parameters, so many concentration doses, various parameters to do the time frame, everything. Eventually, it's very laborious. So through our way of thinking, so we tune the cell, certain pathways that will correlate with specific chemo drugs. So you use this cell line, the whole process is confirmed and we publish. You work together with us, you can get the paper up quite easily. It's just like 3 years of your, it's like 3 years of your PhD. One objective is like you need to do something, but if working with this, actually you can deliver one of our objectives. So that's our simple concept initially. You don't need to do the optimization throughout like 1 year to confirm your experiment is working. But you get this kit, basically you save your time. So this is how we actually kick-started in terms of testing for chemo-resistant cancer or your drug, which is like the activity is not too strong. So we have the series 2, which is the spheroid, it's almost done, and the series 3, I won't mention it. And actually we're working with Taipei Medical University as well at the moment on the prediction model of the machine learning at the moment. So they are transferring around like 2,000 of compounds for us to screen to do the prediction. So these are projects that leads by Dr. Tan Yiwen, so one of the postdocs in our research center now. Okay, so that was like improvement on drug delivery, improvement in terms of like using a specific synthesized cell line that we can do. What else? So we touched a little bit about extracellular vesicles. Yeah. So I think I was introduced to this field by the lady over there, Dr. Wiley. Yeah. So because of her, I touched on the EVs. Yeah. So I think I won't go through very scientifically, but the point is EV is a vesicles that released by the cells, right? So you just think that something like FedEx sending some information to the next cell, maybe the chemo resistant, like let's say we treat the cell, how they form the resistance, how the rest of the cell next generation they know about it. So perhaps because of the EVs, the message inside there could be. So that was the general idea. Why we initially touched on EV, it was because when we do the treatment on the cell, majority of the time we look at the mechanism of action, right? And if let's say you form resistant, if let's say you look at a conventional mechanism, there is no value academically. Okay, research in application, I don't say first. I say academically, we don't have much of value, right? So we're just thinking, during that time, EVs, not too many people are working on it. What if we can check on EVs? Is there any things that inside there can correlate with the risk resistance? Then we found something interesting. That's how the whole thing started during that time. And I think sizes, I won't go through thoroughly. So roughly exosome, we know like around 150. So but extracellular vesicle around 300 nanometer in that sense. Okay, I think the applications are quite wide nowadays. I think we do understood already. And just touch a little bit later on, we touch a little bit on the prostate cancer that we're working on with the European Horizon grant, with Dr. Lee Wai-Ling. And also, I will touch a little bit about the mesenchyma stem cell as well, the projects that we are working with one of the companies, listed company in Malaysia. Okay, with that idea in mind, so resistant, you can see a photo is there, Does that look exactly the same? Yeah. Okay, so with that idea in mind, we test it out initially. Yes, it's true that actually it's working. So with EVs, we can check there's something inside there, message that might play roles in chemo, resistant. Later on, you will see a few more. So I just put a few of them. So we also work, I think this one also just published recently, mainly is on the extracellular vesicle as well. Then we look at the autophagy and also the biomarkers. So that's what we're working on. And if let's say track back, actually we started about some of the concept idea was like 2021. So it took us a few years until the whole project is being done. Okay, I have not much time left. Okay, and we also work with the cancer biomarkers, I think with Sunway Medical Centers at the moment, mainly on the extracellular vesicles that derive from the urine, which correlate with the prostate cancer. So we have projects ongoing, and with European Horizon grant, this project, we are using e-nodes, so electric nodes to check on the volatile components from extracellular vesicles. So it's just that we're using FTIR is one way, another is e-nodes, then combinatory of the way of doing it, then we can see whether we can find the parameters that's suitable for detect the changes to predict the prostate cancer. Okay, this is using some of pilot study that done in then we do the further analysis, and I think recently just published as well. Okay. And similar concept on the EVs, we work with the Sunway TCM centers as well, because TCM, basically you don't have a good markers to detect some of the changes of the treatment. So we also tap into this area and look at the changes. I think we just recently gotten the good data. Alright, exosome, this is led by, definitely the project I shown thus far all correlate with our center here and there, but mainly led by Prof. Rebecca for this context. We work with Supergenic. So it's a listed company that working on the stem cell for clinical setting. And mainly we work on the mesenchyma stem cell. We took out the exosome. But for this project, the applications on the wound healing. But there's other project that took out the exosome is for cancer. So basically it's mesenchyma stem cell, then we take the exosome, then we look at the changes, biofunctionality. So for this context is on the wound. Well, touch a little bit about a single cell stack that I need to do a little bit marketing here. Okay, so the machines is at the 3rd floor of this building, 3rd floor. So mainly it looks like this. And what we actually are doing now, because the machine just commenced around 3 months. So this is the first in Malaysia, and what we are doing now is that we try to set up the different pipelines of experimentation before we really outreach towards our collaborators. So one of it is like, in terms of sequencing, transcriptomic, we doing the single cells, then we will proceed with this. Then lipidomic, we are working with the InBioSys at the moment, trying to set out the pipeline. And proteomic is working with Monash, I think across the road. And in our lab, now we are doing the single cell spotting, and actually just now, Cytovision having talk. Actually, we are trying to do something in our lab using a single cell, then it's spot on the slide, then after that we can do the imaging. And electrochemistry, currently we are using the electrochemistry way to detect the changes as well in the optimization process, because we know that single cells, there's a threshold of detection is very low. So you can't use a conventional bioassays to look at the changes, right? And the real thing is, we have a single cells, we will match with scanning ion conductor microscopy. But at the moment, it's not yet arrived, our technology. And just give you some glimpse of idea. So scanning ion conductor microscopy is like the pipette way to do the imaging. On the same time, you can load the ligand in that pipette, So specific region, you can release it. So in that sense, if let's say we are able to track the receptors itself, and during that time the ligand is being released, you can stimulate the changes of a cell specifically. Okay, then how we going to use it? Yeah, so you can see another Lang Jai there. Yeah, the photo is here. So he's actually hands-on help us to screen the numbers of compound. Yeah, Prof. Mine. Yeah, so through the screening, actually we found some of the compounds that actually synthesized by Dr. Allen, our team member, that actually fall in this category, tryptane, and two of the compounds actually somehow give some activities. So he called me one night saying that, hey, I hands-on doing for you. Now I was just thinking that if let's say I'm female, I will get. Yeah, yeah, but sadly I'm not. Yeah, but he said I need to do something on it. So eventually I said, okay, I will do something on it. And with this data, actually we want to try out on our single cells, the scanning ion conductor microscopy, this one. So what we did, I think 2 days back, our sample arrived in Moscow. So we are using this compound specifically because we know that it will target this receptor. So now we want the whole things to be done there to release and stimulate a receptor. So at the cellular level, we can do precision medicine. Yeah, that's what we're trying to do, just to share. All right. And in our center, we also have synthesized a compound that actually are luminescent. So we work with the biogene, then it becomes the probe. So same concept, like you work with the flow cytometry, your FITCs and so on to look at the fluorescence. But we synthesize compound luminescent, and it was done by our final year student, and I think Dr. Ali, yeah. So we managed to make it happen that the compounds act as the conjugate becomes the probe. So we are doing the patent for this at the moment. And quickly, I need to go through. So Sunway, I need to do a little bit marketing again. So 13 groups. So 13 groups except the education group is non-for-profit. So our center is under education. So we are working closely with healthcare, which is hospitals, as some of you have seen, and we are working closely with Sunway Multicare as well. Okay, I skip through publicity enough. Okay, skip. Okay, skip to here. Important part is Sunway Multicare has 120 outlets across Malaysia. But I think in Malaysia, we do understand that product development, everything could be a problem in terms of functionalities of the product, whether it's really effective or not. So how we actually leverage on the ecosystem of Sunway? We have hospital experts within our faculty, we have a lot of experts as well. Then we have the so-called the pharmacy chain, you know the feedbacks from the customers, right? And university, we have experts as well. So how we link all of them together to cater for the needs of the market. So eventually we formed the so-called the Multicare Pharmacy Innovation Hub. So we get endorsement from the highest authorities of Sunway Group. Then it was announced at the pyramid as well during that time. And how? Because we have some experience in work with the functional food before. So we know a little bit about the formulations, everything. I wouldn't say that 100% experts, but we have the experts from the different field, like pharmacists from the chain and also manufacturers, and we can do the trial, everything's ongoing. Yeah, and jump a little bit, with Dr. Patrick, we also work with certain compounds that are actually good for skin initially, but you know that Malaysia is the manufacturers for the glove. Then we use that compound do as a coating inside the glove. That's how the whole thing started, and also we venture into the radiation protection, those PPE in the hospital normally using lead. So we found the compounds actually it can seal off the radiation, but lighter in the weight. Then eventually we won the awards for Nuclear Agency as well. And I correlate a little bit why we work on this. Actually, we are meant for our space project. Yeah, so it was sponsored by a Japanese company fully, and we sent our samples up to the space, April. And why? Why we work on this? Mainly because we want to create the material to shield off space radiation that will damage the medicine. Yeah, so we send it out, and we will send again by next year for longer exposure. Patent filing is in process now. Yeah. Anyhow, I need to do some publicity, part of the European Horizon grant. So I think in Monash they will conduct it, and our center will provide the single cells training. So the whole team, I think 6 countries, correct me if I'm not wrong. Yeah, 6 countries across the globe that part of this project will be here. So mainly we work on the extracellular vesicles on the volatile components as a biomarker. Alright? And if let's say interested in, I think some of you might visited our center before. I think we have a very comprehensive setup. I think the university thus far invested just equipment is more than 13 million to be safe to say. Yeah. Anyhow, that's about it. Thank you.